Assalamualaikum and hello guys. My name is Anshan Abdullah Halim and I will tell you guys about cloning technology. There are gene cloning, therapeutic cloning and reproductive cloning. Among these three types of cloning technology, the reproductive cloning is the most controversial cloning technology that produces the whole individual with the same genetic. So, how is this cloning can be done? Firstly, researchers remove a matured somatic cell such as a skin cell from an animal that they wish to copy. The DNA was added from the somatic cell to the empty egg in two ways. In the first method, they remove the DNA containing nucleus of the somatic cell with a needle and inject it into the empty egg. The second method, they use an electrical current to fuse the entire somatic cell with the empty egg. The egg then allowed to develop into an early-stage embryo in the test tube and then is implanted into the womb of an adult female animal. Ultimately, the adult female gives birth to an animal that has the same genetic makeup as the animal that donated the somatic cell. This young animal is referred to as a clone.
Human cloning has been used to produce early embryo, making a significant step in medicine. The clone embryo were used as a source of the stem cells, which make new heart muscle, bones, brain tissue, or any other type of the cell in the body. However, researchers said other sources of the stem cell may be easier, cheaper, and less controversial, but opponents say it is unethical to experiment to human embryos and have called for a ban. Stem cells are one of the great hope for medicines, being able to create new tissue, might able to heal the damage caused by the heart attack or repair severed spinal cord. Embryonic stem cell research has repeatedly raised ethical concerns, and human eggs are a scarce resource. This has led researchers to an alternative route of the stem cells. The technique takes the same sample of the skin cells but convert them using protein to induce pluripotent stem cells. There are still questions about the quality of the stem cell produced using this method compared with the embryonic stem cells. Opponents of the new technique argue that all embryos, whether created in the lab or not, have been potential to go to become a fully fledged human, and as such it is morally wrong to experiment on them. They strongly advocate to use the stem cells from the adult tissue.
So, let's talk about the ethics of cloning. When we ask people about cloning or clone human beings, the overwhelming answer is no. But when they are asked whether we should do somatic cell nuclear transfer for genetic treatment of genetic disease, they will say yes. Well, it's the same things. Whether we are talking about cloning or somatic cell nuclear transfer, both are the same. I think the cloning of human beings are the critical issue nowadays. What scientists do with research cloning is they will harvest the stem cells from the embryo and as a result, the embryo is destroyed. Scientists would use them in research with the hope of finding cure for various diseases or gain greater knowledge of early human development. So, how should we evaluate this procedure? Is cloning ethical? The answer is obviously no. Most people are opposed to reproductive cloning, but some will accept the advantages of research cloning and say we should go forward with that.
In fact, in the United States, there's been a couple of states that have passed laws that allow for research cloning but prohibit reproductive cloning. And this is interesting and tragic because what happens is you allow the creation of a human being through cloning, but the human being must be destroyed. Imagine that. This is a law in the United States. It's not a federal law, but in certain states that allow research cloning that requires the destruction of that human being. This should not happen. So, the first and most basic objection to human cloning, to research cloning, is the destruction of human embryos that are created simply to be destroyed. Now, when it comes to reproductive cloning, there are still problems with risks on human life and the destructions, because in animal trials, like the great success of Dolly the sheep, which was 2 decades ago, well, the success came only after failed attempts, well over 200 failed attempts at cloning before that successful attempt. And when it comes to human beings then, are we really ready to allow for the test of so many human embryos? Let's think about it.